Good morning, everyone, or good evening, depending on where you guys are coming in from today. This is John Heerdink. I'm the founder and managing member of Tribe Public, and I'm excited again today to have Dietrich Stephan, founder CEO of NeuBase Therapeutics. NeuBase trades on the NASDAQ, symbol NBSE. I'm proud to be an investor in the company, been in since the company went public. As I participated in a round, and I've added an investment since. Again, thank you all from the tribe that come in from around the world, and I look forward to hearing from you and after this call. Remember, you can send, if you already haven't, send your questions to me via the Zoom chat now, or if you want to do it via email, I'll check that and do my best to look over at my other computer to do so. But I'm going to allow Dietrich to introduce himself again. For those of you that haven't met him, and at the same time, please do send your questions. He's going to do about a 15, 20, 30-minute, depending on presentation, how quickly it goes. And he's had some exciting data that he's going to share with us that came out at the end of last week, and I think will accelerate his company and platform as he moves forward. So Dietrich, thanks again for joining us today. Maybe just say a couple words, if you will, to get us introduced, and then we'll get going.
Of course. Thank you so much, John, for the opportunity to present to your colleagues today. It's always a pleasure to be with you. And again, thanks for being a wonderful partner from the beginning as an investor in NeuBase. My name is Dr. Dietrich Stephan. I'm the chairman and CEO of NeuBase Therapeutics. What I'd like to do today is present an overview of the company to you so you can really put us in the macroeconomic context. Tell you about the exciting data that we just delivered to the public markets last week that we believe proves that we can develop the therapies that we have claimed we can develop and do so in a scalable way. And finally, describe to you why we believe now is the right time for our company from an investment thesis perspective. So our goal is to make a significant impact on the lives of people who are suffering and dying needlessly around the globe from innumerable different human diseases. We've built a rigorous scientific foundation for the company in the short time that we've been in existence, about 18 months, through methodical investments into our platform technology, which, as I'll describe to you, is completely unique and has advantages over other companies in this small group of precision genetic medicine, as well as strategic investments into our team. And I'm really quite proud of the team, and I'll give you some examples of this world-class group that has come around the company. And together, investments in the technology and the team are now propelling us forward toward the clinic and the marketplace in our first two indications. And behind that, we are broadening our pipeline into other disease areas. So the highlight for today will be new proof of concept data that proves that this first-in-class platform can deliver. And that proof comes in the form of injecting one of our drugs into an animal model, which has been modified genetically to perfectly represent the human disease, and that disease is myotonic dystrophy type 1. So showing that we can resolve the causal deficit in this animal directly maps onto our ability to do so in humans because it's the identical mutation that causes both diseases. These are the topics I'm going to briefly touch on over the next 30 minutes or so, and John, hopefully we have 30 minutes today together. I'll try and expedite through some of the contextual components of the talk, and then happy to take any questions with the goal of wrapping up no later than 12:15 p.m. Eastern time today. I always like to start with some takeaways. So what, just to begin with and to end with, why is this an important company and why is this a good investment opportunity, in my humble opinion? We've produced and disseminated data earlier on that validates the foundational first-in-class platform. And what that data showed was that we can spit out a new type of therapy called an anti-gene, which can home in on a misbehaving gene in the body of a patient and neutralize it before it can form a misbehaving protein and cause disease. And so we showed that after single dose injections into non-human primates, which are, as you know, the most closely related species to humans, that our drugs can get everywhere and access every cell and tissue in the non-human primate body, and do so at therapeutically relevant levels. So why is that important? If you can't get to your target, you can't neutralize it. And that data set, which we disseminated in March, proves that with an easy to take shot, we can get our drugs to where they need to be, even into the brain, which is one of the grand challenges of drug delivery, as all of you know. It's what's hampered our progress in central nervous system diseases. And we have something special in our ability to get in. We also showed that our drugs are very specific for just the misbehaving gene, and they can turn that misbehaving gene off without impacting normal function across any other normal gene. And that's important. You don't want to be switching good genes off. That could cause more damage than you're trying to alleviate. And finally, we showed that our compounds are well tolerated in both lower order animals, rodents, as well as higher order species like non-human primates. So taken together, we have a single unified platform that can turn genes and proteins on if we need to boost their levels to cure disease, off if we need to get rid of a disease-causing gene or protein, and even change a gene's function. So together, we have attributes of companies like Ionis now Alnylam, which only silence misbehaving genes, or companies like Moderna and BioNTech, which only turn genes on, or companies in the genome editing space, which can change protein functions. So here we have a company that can do all three and address any mechanism of causality with good biodistribution and good tolerability. So that's the upside opportunity. We're not going to get there tomorrow. This is a mid to long-term story, but we do believe that for the long-minded investor that's looking for a transformational platform technology, that we fit the bill. Got to start somewhere. So we're starting in Huntington's disease and myotonic dystrophy, both of which are horrific. There's no cure for them. They're relatively common, and they're devastating. And today, what I'm going to talk to you about is an extension of that foundational work, so that we can dose an animal that suffers from myotonic dystrophy, again, with a precise human mutation that patients suffer from, and with a single dose injection that we resolve the causal process or the disease-causing process in this animal model. And we don't, like many of the other attempted therapies, actually make the situation worse by getting rid of a critical protein in this biological process. So I'm going to tell you about the data, but I definitely want to anchor you on the macro context of this story. So what I like to do is remind people that we're in the midst of a transformation in the pharmaceutical industry, one in which we move from historical trial and error based drug screening for active agents that stick to disease causing proteins or cells and make them better. And if we find some of those historically through trial and error screening, it often takes a decade and billions of dollars to chemically engineer those compounds so they work and they don't stick to good things. So we're moving from that old way of working in this industry to a new way of working where we can actually go upstream of the protein and target a mutant gene with something analogous to a heat-seeking missile. And most, if not all, diseases are genetic. And this affords us the benefit of knowing the causal event behind many diseases. But more importantly, we know the precise sequence of the target and its precise atomic structure. We don't have to guess, and we don't have to do trial and error screening. Thus, we're using nature's own digital information encoding schema, the genetic code, to custom design potentially highly precise and more tolerable drugs that correct gene function. So how do we do this? We encode the sequence of the gene we wish to neutralize right into our drug. And that's into this biologically inert scaffold that we use. And if we can do this correctly, we only engage with the broken, misbehaving target and no other target in the genome or transcriptome. And thus, there's the potential to have very clean drugs. So this conceptual approach, coupled with the advantages of our technology over other approaches, promises to have a large impact on suffering and death globally. And as with any platform technology, we believe the cost of development will decrease over time. And we see evidence of this in the marketplace with some of the companies that I mentioned that were a little earlier out of the gate with first generation platforms. The better they get in terms of understanding their platform, the more drugs they output, the faster they can output them. And we believe we have that scalability implicit in our platform as well. So cost goes down, rate of development likely goes up, and the failure rate over time likely decreases. All right, let's get some data so you can actually touch and feel how these work. So we're going to talk about myotonic dystrophy type 1. It's a dominant genetic disease. So about 50% of a patient's offspring will have the disease. And its prevalence is about 5 to 12.5 per 100,000 in the general population. Again, fairly common for what's called an orphan genetic disease. There are 3 forms of the disease, and they're characterized, the severe form is characterized by myotonia, severe muscle weakness at birth, often accompanied by respiratory distress, intellectual disabilities, and early death. The second is the classic form, which is characterized by myotonia, muscle weakness, and wasting, often with cardiac conduction defects, and adults may become physically disabled and often have a shortened lifespan. There's also a mild form of the disease with more subtle myotonia. These patients generally have a normal lifespan. And taken together, these folks on average have an increased healthcare spend of tens of thousands of dollars in excess of their unaffected counterparts per year. DM1 is caused by a mutation in a single gene called the DMPK gene, and I show that at the top right of the figure. The mutation itself is what's called a trinucleotide repeat. In this case, it's 3 letters in the genetic code, CUG, that are repeated over and over and over again, and that's aberrant. And so when it's in its expanded form, it actually causes the disease. That mutation is in the very end of a gene called the 3' untranslated region, and it actually doesn't cause an abnormality at the protein level. It's copied into what's called an mRNA or messenger RNA, and it forms an aberrant hairpin structure. And that's what you see in the middle of the slide, where you have a single strand of messenger RNA forming a stem loop structure. And that aberrant hairpin attracts proteins to stick to it in the nucleus and causes aggregation. And that's really the root cause of the disease. This aggregation eliminates the ability of those sequestered proteins that you see in red to go do what they're normally supposed to do in the cells, and that's regulate normal processing of transcripts for other genes, a process named splicing. So in addition to widespread splicing defects that cause, for example, myotonia, you also have a situation where you don't make enough of the DMPK protein because half of the RNA is stuck up in the nucleus. And that's called a haploinsufficiency. And so 50% is not enough of that normal protein. And that has been shown, at least in animal models, to cause serious cardiac conduction defects and problems with learning in the brain. Two things that I described you also see in patients. And so this primary mutation causes two causal events that result in the disease itself. There are several strategies being employed to cure this disease by the community. The first is to get rid of that DMPK mRNA, just degrade it so that you release those aggregates in the nucleus, and those red proteins that I showed you can go do what they're supposed to do in this cell. And that strategy works. It rescues splicing. The problem is you degrade that protein, and there is evidence out there that you want to keep that protein around for normal, healthy functioning. And so we're not an advocate of that first strategy. We actually employ the second strategy listed here, which is we're designed to disrupt those aggregates, rescue normal splicing, but not to explicitly degrade this important protein called DMPK. So we believe that we have a best-in-class solution that promises higher tolerability and lower side effect profiles for patients in the marketplace. And these are the tissues in which normal DMPK remain turned on in healthy adults. Skeletal muscle, regenerating muscle, at the neuromuscular junction, in the heart, and in the brain. And so by turning those off, we risk exacerbating this disease. Okay. All of the in vitro and in vivo data you're about to see here has been conducted with one of our compounds called compound A. What you see here are cells from real human patients. So these are human patients grown in a dish and dosed with compound A. In blue, on the right-hand side of the slide, you see that target RNA transcript that we're aiming to fix. And these are in blue, you see the levels in disease cells. So they're excessively turned on. In black or gray on the right-hand side, you see the levels of that RNA in healthy cells. And so within 24 hours of dosing with compound A, we normalize the amount of that mutant RNA and show that we engage the target. This also shows that our drug rapidly crosses cell membranes, gets into endosomes, escapes from endosomes, gets into the nucleus, and normalizes this classic overexpression of the DMPK transcript. So in one synthetic compound, we're able to traffic and normalize. In addition, when looking at several other known transcripts that are inappropriately spliced in patients with DM1 or myotonic dystrophy type 1, these transcripts are named muscle-blind 1 and 2. We show that within 2 to 5 days, we normalize their processing in the cell. Remember I told you that generalized missplicing is what causes patients to have myotonia. Here, with these two marker genes, we show resolution of normal splicing beginning at day 2 to 5 and continuing to mature over time to day 9, where we almost completely normalize that missplicing. And if we expand the aperture and look at all misspliced transcripts between diseased patient cells in blue and healthy individuals in gray at 0, we see that at 5 days, we get mass induction of normalized splicing across these more than 100 different transcripts, and that phenomenon progresses towards complete normalization over time. Here's a another visual of that. So each of the columns here Across the x-axis going out to 200 is a single human gene that's inappropriately processed in patients. In blue is a patient cell with a disease. In gray at 1 is the way that that transcript is processed in a healthy individual. And what you can see in every one of these cases is with treatment in green, we get movement towards normal processing. And here are a couple of examples of specific genes that, again, at day 5 have complete normalization of this generalized causal defect in cells from real world patients. This is a big deal because we show that we can address causality and restore normal healthy functioning in cells. Okay, that's all well and good to do in cell culture, albeit it's important to do it in human patient-derived cells. Now let's make sure that you understand that we can do this in animal models with the same genetic defect. And this is more complex because you can't just pour it onto the cell that's defective. You actually have to give a shot to these animals, and the drug has to circulate through the body, find the target tissue, get through the extracellular matrix, which is a lattice work that embeds these skeletal muscle cells, get through the myofiber into the nuclei and so forth. And so it's a non-trivial and complex delivery challenge. And we used the gold standard animal model for myotonic dystrophy type 1. I won't belabor this. It's a model called the HSA-LR model. It was developed by our collaborator, Charles Thornton in Rochester. And he inserted the same genetic defect into this mouse that causes the human disease, namely a CUG repeat. And after a single dose intravenous injection of compound A, when we harvest the muscles of those animals after 24 hours, we see normalization of the target mRNA, again in green, relative to healthy, untreated animals. So again, just to outline that process, because this is a key component of the differentiation of our platform. We have a fully synthetic drug. We don't have to couple it to antibodies or big honking peptides and do all kinds of complex chemistry. It's a single synthetic scaffold that has delivery technology baked into it, injected IV. We have a nice circulating half-life in the blood. We can home in on skeletal muscle, get into skeletal muscle, and engage the target, which is what you see here. And when we look at misregulated transcripts globally in those tibialis anterior skeletal muscle from this genetic mouse model, what you see is that we also rescue broad-based missplicing. Again, the hallmark feature of causality of this disease. So in blue, you have an animal with the disease. In gray on the left, you have a normal animal. And in green is broad-based correction of the splicing defect across a substantial number of genes. Here you see the same graphic. Again, each column is a single gene, and you see that we move from the blue inappropriately spliced transcripts in each column to the green, where we normalize, and in many cases have complete normalization of missplicing. And you can see that here. And so, for example, if you pick any one of these, let's just look in the top left here. As an example, what you see in blue is a gene named GM229394. It's a mouse gene that's inappropriately spliced in diseased animals in blue. As soon as we add the drug and wait 13 days, you see that we get normal splicing occurring at the same degree as in a healthy, normal animal. And this story continues for gene after gene after gene. These drugs are well tolerated at this effective dose, and these are now positioned to move forward in the development program. Finally, and importantly, it's critically important to remember that we also don't want to completely degrade that transcript, because it's important. The protein product of that mRNA has functions in the brain and skeletal muscle and heart. And what you see here is that after dosing with our compound A in human cells, we do not further deplete that protein. And we take this as a good sign that we potentially have a best-in-class therapy for these patients. Okay, so we believe that we've presented data to you that shows that our platform can output effective, easy to take, and well-tolerated therapies. And now I want to touch on a forward look as to how we get from here to a world where we have substantial impact. And I'm just going to race through this, but this is a modular platform. It's actually made up of 3 buckets of parts. The first is the delivery technology, which I alluded to. This allows us to take any payload and deliver it systemically and allow it to get to where it needs to be. We can also add enhancer modules to these, which allow the drugs to do things like get out of endosomes better or go to the nucleus versus the cytoplasm. And we like to think of these as enhancer delivery technologies, which we can append to our delivery stack. The workhorse of the platform is the synthetic scaffold. This is the second bucket of parts, and this is what is very unique. It allows ultra precise engagement with its target because it's semi-rigid and it cannot bend around a mismatched target. It's very high affinity. So once it engages, it generally doesn't come back off. And it's neutral in charge, which is important because it doesn't trigger a lot of the clearance pathways in the liver that stymie folks that are using anionic-based scaffolds. And so we like to think that these have fewer of the limitations of other genetic medicines that are in development today. And finally, the last bucket of parts are the nucleobases. And these are the telemetry system, if you will, of these heat-seeking missiles. Not only can we use normal ACs, Gs, and Ts, but we've got a suite of engineered nucleobases that further improve precision of engagement to targets, which is important because off-target engagements cause issues. You don't want to turn good genes off inappropriately. So those are the platform components that we believe allow us to snap together various drugs fairly quickly. Again, once we get further subsequent platform validation components in place, such that next calendar year, over the next 12 months or so, we will be doing the IND enabling toxicology work for our first human studies. And we do have 2 programs that are moving in parallel forward. We presented data to you today on myotonic dystrophy that's driven off of the PK data we presented last time in March. Huntington's disease is running in parallel, and we look forward to updating everyone at our research day that's coming up in the first half of 2021. And I would like to remind you that we have a crackerjack team of heavyweights around the company. For example, recently we just recruited Kurt Bradshaw. He was the most recent chief science officer of Arrowhead, another company in this small group of companies in our space. He helped grow that company to a $7 billion market cap. He's filed 12 INDs from basic research into first in humans and has participated in another 24 clinical trials. We recently also brought the inventor of peptide nucleic acids, Peter Nielsen, on board. He first described these in Science in 1991. And of course, continue to work hand in hand with colleagues like George Church and Sam Broder. And then it's really important to make sure that all of you recognize that this is a pharmaceuticalization, if you will, of a lot of academic research work that has happened historically in university labs. And this is not a science project. There are a cadre of drug developers around this that have collectively brought dozens of drugs to market and have the perspective and experience to ensure that we do all of the right things, including creative problem solving, because this is biotech, to maximize the potential of this platform. And that really is what we're working to do on behalf of all of our shareholders. And so I'll stop there and just again want to remind you that we've moved from the platform validation phase of our life in the short 18 months that we've been in existence to now moving our programs forward towards the clinic. And that's our focus at this point in time. And we've got an exciting pipeline behind that. So happy to take any questions, John, and thank you again for the opportunity to present to you today.
Absolutely. Thank you again, Dietrich. We've got a couple questions here already lined up, and I'm going to remind anyone that's online here is that you can easily just type it through the Zoom chat feature if something comes to mind as addressing other questions. I'll start with in no certain order, but I've got a couple questions. One is, can we, and I think you've already addressed this, but let's just make sure we get it covered. It says, can we expect to see additional data for compound A in different tissue areas outside of the tibialis, I don't know if I'm saying it right, anterior muscle, for example, heart, diaphragm, liver, kidney, and quadriceps muscle?
The answer is absolutely yes. This specific animal model, again, the gold standard of DM1, the HSA-LR model, only expresses the disease in skeletal muscle because of the way it was made. And so it will not manifest the symptoms of disease in smooth muscle, cardiac muscle, or CNS. And we can get into the technical aspects of that later on, but it's driven off of a human skeletal actin promoter. And that's why you don't manifest in those other tissues. That being said, of course, we believe our broad biodistribution has advantages in getting this drug to other tissues that are affected in humans. And that will be a component of the work that we do over the next year before we get into humans to really understand what we should expect from our efficacy trials as we go into humans.
Okay, thank you. The second question is, you demonstrated retention of DMPK levels in cell models in vitro. Were DMPK levels also evaluated during an in vivo studies and maintained, and to what extent, if they can be measured?
Yes, so we have looked at DMPK protein in the animal model that we presented. The challenge with that model, and again, I hate to get into nuances of the model, but the transgene that's causing DM1 in that case has this CUG repeat in the 3' UTR. That gene is called the human skeletal actin gene, and it's knocked into that animal. And so in the animal model, rather than looking at retention of DMPK protein, because there's nothing wrong with the DMPK gene or protein in the animal, we actually looked at the human skeletal actin protein levels. And there we see what we wanted to see, which is that the protein levels are not further diminished, which should not exacerbate the disease in that animal model. And we think directly maps against what we will see in humans, which in that case will be retention of DMPK protein levels. So a little bit of a apples to orange there, but we're seeing exactly what we expected in the animal model.
Okay. It says, you mentioned that NeuBase will be taking a one compound at IND enabling studies in 2021, with first humans likely occurring in 2022. How should we interpret this? Has the HD compound had setbacks or unexpected results? If not, how will NeuBase decide which compound to take forward, and what criteria you're focusing in this decision? I think you've addressed this, but could you sum up that?
Yeah, no, thank you for the opportunity to address this because I haven't been as clear as I need to be on this topic. So not only has the HD program not slowed or in any way sort of had any negative forward motion. In fact, it's the opposite. The DM1 program has accelerated and performed better than we had hoped. And so we wanted to make sure we get that data out the door and illustrate to the entire community that this first-in-class platform performs exactly as we had hoped in terms of precise engagement with a mutation, addressing causality, and nice tolerability. So I would think of both of these disease programs moving forward in parallel. The reason that I am being a little conservative in guiding to one, getting into and through IND enabling toxicology work and into the clinic is because we've just hired a brand new chief scientific officer, Kurt Bradshaw, and he started mid-December. And what I want to make sure we do as a company is fully get him up to speed. That's not a long process, but he needs to deeply understand what we're doing and then own the timelines. And so what I've done is project forward a little more conservatively than perhaps folks are used to allow Kurt that time. And I believe firmly that this is one of those situations where it's a go slow to go fast. This guy has been proven to underpromise and overdeliver again and again and again. And I want to set him up for success and not cause him to get into any sort of mindset where he's being tethered to timelines that he doesn't own. And so that's what you're noticing here. It is not a reflection of any negative drag on the Huntington's program.
Thank you. You spoke previous conversations that we've had both with the tribe and us and me otherwise and investors about corporate development. And you had a financing after the first set of data earlier this year that bolstered the balance sheet. And I believe might have accelerated some discussions of corporate potential corporate partners. Can you speak to that? One, the financing, how you're set up for the next couple of years, and at the same time, can you speak to any corporate development discussions to the degree that you can at this point?
Yeah, thank you. So we're in a 9/30 fiscal year, and so you'll see our K shortly. We have talked about our cash runway through early 2022, which allows us to hit several important catalytic milestones. I think most notably IND filing and in one program. And so we have a nice cash runway. Just for context, I think it's also important to just take a half a step back. And so this is a relatively new story, not only to you all, but to the institutional investment community. So our CFO, Sam Backenroth, and I and our board have been doing a lot of NDR road work, socializing what we're doing to fundamental institutional investors, as well as potential pharma partners. And the trigger that everyone has articulated, well, that most have articulated that would allow them to engage with NeuBase is what I just presented to you today: in vivo efficacy in one of our two first indications. And so we just delivered this last week, and I do believe that going through an organized process on the back of this will allow us to engage with a pharma partner on good terms. And so what I mean by that is we've proven that this platform can work, and I think we've hit the sweet spot of not only their interest, but on potential economics around a BD deal. So As you can imagine, doing those deals too early likely gives away value. Doing them, with this data in hand, I think positions us nicely. And so we've previously messaged that we're hoping to announce a pharma partnership within 1 calendar year of our last transaction. And I'd say we're tracking nicely against that. I mean, could it slip a couple of weeks or a quarter? Yes. But remember, this is biotech. And my intent is to build a dominant company without unnecessarily giving away shareholder value in the short term. And so just please work with us on nuances of timing. It will happen. We're deeply engaged with 6 to 9 big pharma partners, and I would say there's interest in what we're doing.
Fantastic. One of the things I think people should remember here is, and maybe you could speak to this, is that this isn't your first rodeo. You've had a number of exits, and maybe you can confirm those exits successfully as you develop through your career. And that experience, how does that experience give value to what you're doing today? One last thing I would remind everybody, many of you that I see that are on the call today have either invested or heard me chirp, if you will, about Fate Therapeutics, another company on the platform, symbol FATE. And this is a company that similarly, very low valuation when we started with the company. And by the way, just recently, a couple of weeks ago, if you hadn't noticed it, they had, with 4 patients, a phase 1 data readout that excited the world. And if you haven't noticed, the stock has gone up from whatever the 100, 200 million, basically where NeuBase is today, is now hit a high over $9 billion in valuation on relatively early research. It's my belief there's going to be a great deal of value created if he successfully pushes through, and even in the early stages of this, of his company. And I think that's what's very interesting to me, because the world is looking towards these platform opportunities to derive value and progress for science. So anyway, if you wouldn't mind going back and talk about your exits and a little bit about that, that might be helpful as background.
No, thank you, John. And as you know, I generally don't like to talk about myself, but I've had a first career in academia where my last posting was as chairman of genetics at a top 5 medical school, and ran a department that, for example, was instrumental in co-developing Genzyme's first enzyme replacement therapy and a number of other, I believe, in first-in-class type of therapies. In parallel, have founded or co-founded 14 biotech companies, loosely split evenly between molecular diagnostics and targeted therapies. Those have all been backed by VCs from predominantly the San Francisco Bay Area, including Sequoia Capital, which has backed 4 of those. John Doerr and Brooke Byers at Kleiner Perkins, Larry Page at Google, or David A. Ventures, and Peter Thiel. Most recently, you probably saw an announcement yesterday around a financing of a company that I was involved in helping to put together named Peptologics. I do remain as chairman of the board there, but I mentioned that because Founders Fund and Point72 and Thiel Capital and a number of others are in that Series B financing that was just closed. And so I come from the world of marquee VC-backed biotech companies. I made a conscious decision not to take that route with this company because I believe that management would not be fully incentivized had we gone the traditional route to do the heavy labor that's required in building a company like this. This is a platform company with unlimited potential, but it's a lot of hard work. And So we've created dynamic internally within the company that all of our team members are fully incentivized with upside. I'm the #1 shareholder in the company. And so if any of you have any doubts about my motivation, I'm fully incentivized, and I have purchased shares in both of our financings as well as in the open market. So something important to remember. We also had the luxury of, because of the way we went public, of handpicking our own board. So these are all people that have actually done this before. These are not passive investment managers. And that's a critical operating nuance for anyone that's ever run a company, that you can lean on your board for help and have them be fully aligned, as opposed to having to manage a board with misaligned incentives. And so this company is fully sort of put together, aligned beautifully. And quite frankly, it's the best team I've ever worked with in my life. And so I'm proud of the company and the team. And long answer to short question, Don. Sorry about that, but you got me on one of my favorite topics.
No, it's one of the things that excited me about the original, when I first heard your name and heard NeuBase back in 19. And, I completely agree. I see that you build a foundation, you've been adding it smartly with the add. The new CSO and your SAP members that you've added here. I can see how you're methodically building this company. It'd be interesting to see when the world realizes what you have, but I think it's we're not too far away from that, and we'll do our best to let a few folks know about what's going on. But I guess a follow-up, if you could sum up, if will, this sort of milestones. I don't know if you have that in this particular presentation, but what do you see, what have you put to the market over the next year, quarter, what have you broken it down, first half, second half? Could you just review that with us, and these value generating milestones you're looking to achieve?
Yeah, so our trajectory is to build a rock solid foundation in terms of our platform and our team. And so you'll continue to see that harden over the next year. We're going to announce some, I think, extremely exciting new team members in 2021, and that will speak to our ability to continue to access the capital markets and do business development at the highest levels. So that's been something that's we've been working on methodically to make sure that we nail it. Out of that solid foundation, you'll see these programs begin to emerge. The first two have been named. And again, the level of granularity we're giving right now is that one will be in IND enabling toxicology work. The other is not far behind it. That's the way I would think about it, with the goal of the stretch goal of late 2021, early 2022. Let's say early 2022 of being in the clinic with our first program. Behind that, though, we do have a pipeline emerging. I was hoping to talk about that here in the fourth quarter, but again, out of respect for our new CSO, I do not want to get out ahead of him. And what we have planned to do instead, and I understand that that's people were excited about hearing about it. You're definitely going to still hear about it. We're doing the work in the background. We're going to unroll that at our research day, which we're planning for the first half of 2021, where we're really going to fully open the kimono and allow people to understand all of the nuances of the platform, the two programs, and the pipeline expansion. And so that's something we're excited about. Is
there a target date, or you're still working on the date for that research day?
We're still working on the date for that.
Okay, thank you. And I guess you're going to be busy, as JP Morgan is coming up here in the mid-January again, even though you won't be coming out to see me in San Francisco. I'm sure those meetings or calendars, your card is probably full. kicking off the year. So we're happy to get you in here before the new year because I'm sure you're going to be super busy. But on the stock side of things, you've got a number of analysts that are covering you at respective banks. Can you speak to that? Who that is? How many that is? Is there a target price range that you could throw out to us? Because many of the people don't have access to that research. Some do, some don't, depending on who's on the call here.
Of course. I usually have a slide on that, so I apologize. So our market cap is roughly 200 million. We believe our stock is still a value-based stock relative to some in our peer group that are a half step ahead of us. I referenced the recent IPOs of Dyne and Avidity trading at about $1 billion market cap. If you look at their data, it's near identical to the data I presented you today. And so that's why I think this is a timely story. We have 6 covering analysts, Oppenheimer, RBC, BTIG, H.C. Wainwright, Guggenheim, and I am ashamed to say that the 6th one is eluding me right now, and it's
Is HCW covering you or?
HCW, yep, H.C. Wainwright is there. I apologize, I will get you to, and the price targets are between 15 and 18, okay. And they're all a buy or outperform rating.
Gotcha. And we're trading right around $8 right now. I know we've been oscillating between, sort of that $8 range, $9 range. And so it's interesting. And I'm sure others will start to come on since you've got the data out. I would guess that's in the works too.
And it's interesting, just one comment, while we haven't been impacted from an operational perspective through the pandemic, we have, and really the team has gone to heroic measures to stay healthy and ensure that we can continue to operate. Our first data set was released publicly at the very beginning of the COVID pandemic, and in my humble opinion, largely lost in the midst of people being focused elsewhere. But the highlight there was that we could get our drugs into non-human primate brains after a single dose administration, which is a big deal. And most recently, last Wednesday, we announced this data almost simultaneously with the Pfizer approval of their vaccine. And so in a way, we have been negatively impacted from a story perspective with COVID. And so just a little bit of an uphill battle there, but we'll get through it in the new year.
Sure. By the way, one of the guys on the line here, Randy out of Daylight, Indiana, where I'm originally from, as you all know, runs a biotech fund. He said it's Chardon that's covering.
I'm so sorry. Yes, of course. Thank you.
Yeah.
Hello, my friend Kay at Chardon.
Yeah, thank you.
Yes.
I guess I lost my train of thought here. One of the last things I wanted you to cover is, I guess, reconfirming the insider ownership is quite significant here, and you've got some interesting investors. David Einhorn at Greenlight originally bought around 10% of the capital the company when it was going public. Can you speak to sort of who's invested now? Anything marquee that you can fairly point to, and again, kind of reconfirm what your ownership is today?
Yeah, so insiders management own probably 30 to 35% of the stock. My ownership is a little over 20%. Greenlight Capital is our largest institutional holder. They came in both of our financings. And so we're proud of that. And in our last public offering in April of this year, we had several fundamental investors come in, and some of those are publicly disclosed and some are not because they don't meet the reporting threshold. But we're pleased that we've begun to institutionalize our shareholder base with folks that deeply understand the science. And And I think, a validating pharma partnership will be a great next step to complement them.
Agreed. Thank you. I think we're going to call it a close today, and then we'll look to beg to get you back on sometime in the near future. But is there anything you would leave us with at the the close here? And again, I want to thank all, as Dietrich does that, I want to thank all the tribe members for joining, and continue to remind you that if you want to see Dietrich and or others, we have our wish list process at tribepublic.com that's continually going on. Please send us your interest, continue to do so, and thank you for the interest to date that you've added. And we're excited to really help you give this direct corporate access to leaders like Dietrich, and I look forward to hear from your thoughts after the event. I thank you dearly for participating today, both Dietrich and all the tribe members. But Dietrich, could you close us out with any summary about your company?
Yeah, I mean, well, first of all, John, thank you, and thank all of your participants today for their time. It's always a pleasure and honor to participate with you, so thank you. Secondly, we plan on building a transformational company, and sometimes to do that for the long term, you need to build a rock solid foundation and be methodical and systematic and thoughtful. And if I could give you any insight into how we come to work every day, it's that, coupled with truly world-class execution. And then finally, I would just like to wish everyone a wonderful 2021. Hopefully it's a little smoother than 2020. Health and happiness foremost. So thank you all.
All right. Thanks a lot. Have a great day, guys.